head by Dr. Rajpal Yadav of the World Health Organization, looking at the global vector control response strategy from the WHO till 2030. We went straight into the deep end with genomics and proteomics in terms of genetic signatures, allele transfer, proteomics informed by transcriptomics in terms of the transcriptome and the interactions between the arboviruses themselves and the vector. And then a very interesting panel in terms of the ASEAN country Ministry of Health panel. Ministry of Health Indonesia, Malaysia, and Philippines all backed our World Dengue Day campaign live here at BITES. So that's a really big deal for us. We're a tiny organization, and we've been working very hard on that. And we'd urge you all to sign our petition. We've got over 25,000 signatures asking the WHO for a World Dengue Day, so that's perfect. Last session was fascinating in terms of built environment, the intersections between vector control, rapid urbanization, disease prevention with a focus on snake bite, and then looking at how to disease-proof the built environment and the massive opportunities open there. Some of the earlier presentations touched upon insecticide resistance as a major issue. Certainly the ASEAN countries mentioned it. In fact, some of the genetic signatures work and the genomics and proteomics session also mentioned that. And certainly in the global vector control response alignment to that particular WHO 70.16 member state ratification of that, it was definitely part of that as well. So we wanted to kind of have that in terms of challenges and solutions to insecticide resistance. But a very interesting first speaker, Dr. Rafi Rahman. He's from the Laboratório de Fisiologia de Controle de Artrópodes, vector arthropods, basically at the Fiocruz in Brazil. My Portuguese is not very good, but from Fiocruz. And he's actually looking at insecticide resistance in Pakistan, a kind of joint study there. So I'm going to hand over to Rafi. I'll just bring Rafi up there. Assalamu alaikum wa rahmatullah. Rafi, I hope you're doing well. And
can you listen and see me?
We can see you perfectly. We can hear you perfectly. The light is good in Brazil. So
yes, although it's raining, but it's fine.
Oh, fantastic. It's usually raining here in London, but it's the kind of, we've kind of swapped things around. In this COVID age, everything is the other way around. I'm going to back out, and we'll have a round of applause for the previous session and for you, Rafi.
Just give me a second so I can share my screen and you confirm that you're seeing, okay?
No problem. No problem. We can do that. We can. That's about as much tech as I can handle. So yeah, I'll definitely be happy to do that. Yep, it's on there. If you could launch the slideshow so it's full screen, yeah, then I'll back out. Perfect.
Okay, should I start now? So hello everybody. I hope everything is all right on the technological issues. So my name is Rafi Rahman, and today I'm going to be talking about insecticide resistance status and selected mechanism in Aedes aegypti and Aedes albopictus populations from Lahore, Pakistan. As Kamran introduced, my name is Rafi Rahman, and I have recently concluded my PhD in parasite biology from the Oswaldo Cruz Institute and Fiocruz. Thank you so much, the International Society of Neglected Tropical Diseases, for giving me this opportunity to talk about insecticide resistance in Pakistan, right? So as you know, Pakistan is located in South Asia, and Lahore is considered as the cultural capital of the country and is also known as the heart of Pakistan, right? Dr. Soumya talked in details about dengue in Lahore earlier this morning, and I'm going to be discussing about how the vector control activities are going there, not as she talked about as vector surveillance and disease surveillance, but insecticide resistance, right? So there is a saying about Lahore that in my country, in Pakistan particularly, that if you have not seen Lahore, you are not yet born. So there are some good pictures of Lahore. It's a historical city. It is located on a very important geopolitical location. And as I mentioned, that Dr. Somia told about the dengue in Lahore, the first and major outbreak of dengue took place in Lahore in 2011. As we talk about the vector species of dengue, both vector species are present in Lahore, and we know that there is no vaccine or a drug which can treat or cure dengue and other arthropod vectors, so the only option that we have is the vector control. There are a lot of tools for vector control, but insecticides is one which can be used in emergencies and is the practical solution to the diseases like dengue and other arthropod-borne diseases. The insecticides that have been used in Lahore are from different classes, like their pyrethroids, organophosphates, IGRs, or biological agents like BTI. There were some trials of using the predators for the larvae, of course, and adults, but they were not really successful. But the pyrethroids that were used were permethrin, deltametrin, and alpha-symetrin is currently in use. Organophosphates have been used both as larvicide and adulticide. We know Tamifos, everybody who work with the vector control, they are aware of these terms. IGRs like pyriproxyfen and difluconazole have been used in Lahore as well, and BTI also. But there are some complications with all of these agents, and the major issue or the major concern about using insecticides is insecticide resistance. This picture, you can see that there is a cloud of DDT, and the susceptible mosquitoes, they're died, and the resistance are asking the DDT here, but you know, it was from that time, so it is asking to try it, you know. So insecticide resistance is not the issue of Lahore or it's Rio de Janeiro or Sao Paulo or whatsoever. It's a global issue, and the World Insecticide Resistance Network brought together some brilliant scientists which are still working, but they were the World Insecticide Resistance Network brought them together on the same platform, and they published some really great articles. There were 2 international congresses under the platform of World Insecticide Resistance Network, and here they showed that how is the situation of insecticide resistance on the global scale. So here on the left side, you can see the deltametrin resistant population, Aedes populations, are really frequent or can be found in Central and South America and Southeast Asia. There are few studies in Africa, but we can see that there is still resistance. On the right side of this slide, you can see that Tamifos resistant populations can be found in Central and South America and Southeast Asia, and also few studies are reported in Africa. Here is a point that I need to mention, is that there are like no studies about insecticide resistance in Pakistan, right? There are, of course, some studies, but they do not talk about the mechanisms. So the objectives of our studies was to find the status of insecticide resistance in Aedes aegypti and Aedes albopictus in Lahore, and to investigate the mechanisms selected for resistance. So we did larval bioassays to find out if the populations are susceptible or resistant to Tamifos and pyriproxyfen. So for Tamifos, we performed dose response assays. Dose response assays, we use different range of concentrations, which gives us lethal concentrations and subsequently resistant ratios. In the pyriproxyfen assays, we use a single dose, and the assay is then called the dose diagnostic assays. This assay gives us the adult emergence inhibition and life stage transformations. The results showed us that as we increase the concentration, the mortality is increased, right? The point interesting here is that the mortality between this lab's reference strain and the field strain is not significantly different, right? So it means that we still can use Tamifos in the field against Aedes. On the other side, dark green, this dark blue is the pupal stage. So it caused 100% mortality, mainly at the pupal stage. In order to find out the efficacy of pyrethroids, we performed WHO tube test, where we impregnated filter papers with the doses which are recommended by the World Health Organization. we used 4 pyrethroids: deltamethrin, permethrin, ethofenprox, and alpha-cypermethrin. 3 of them, like deltamethrin, permethrin, alpha-cypermethrin, are used in the field, and we use ethofenprox because it is not a type 1 pyrethroid, neither type 2 pyrethroid, but it's a pseudopyrethroid, and we were like curious to find out if populations are susceptible or resistant to ethofenprox. For malathion, we performed the CDC bottle test. We re-impregnated the bottles, glass bottles, with a diagnostic dose of malathion, and we used a very low dose as compared to WHO dose. Like we perform these tests in our lab, we first use lower doses, and then we perform if the populations are resistant to the doses of our lab, then we use the WHO doses. So here for malathion, we use 20 microgram per liter, which is the dose of our lab. Result shows us that here you can see that none of the pyrethroids called 90% mortality. This red line shows the 98% mortality. What does it mean by 90% mortality? It means that there is suspected resistance in this population. But when the mortality is less than 90%, then the resistance is confirmed. So the populations are confirmed resistant to all of the pyrethroids, but on the other hand, both Aedes aegypti and Aedes albopictus are susceptible to organophosphate malathion. Okay, they're resistant. What mechanism have they selected? As the article I mentioned, if the population are resistant, there can be target site modifications, or there could be metabolic resistance. Here you can see that if the population, as I mentioned, is resistant to pyrethroids, either one of or combination of these mutations should be present in the population, or if they're not present, there should be any of these enzymes which are related to the detoxification of the insecticide should be present, should be overexpressed, right? So let's see what's going on. So in order to find the knockdown resistant mutations, we did the carrier genotyping. We performed 3 experiments. We did SNP genotyping, we did high resolution melt analysis, and we did sequencing, right? Here is a schematic diagram of voltage gated sodium channel. All these carrier mutations that I have mentioned, that they occur in these domains. And in these domains, the sixth segment is really important. Like this 989 mutation is found in the loop between S5 and 6, but all other mutations, if it is 1011, 1016, 1520, it's not shown here, or 1530 to 1534, that they occur in the 6th domain. Okay. Result shows that there are 3 possible variants. The HRM differentiation plot showed that there are 3 positive variants. Our sequencing showed us this. What are those variants? So this green variant, this green group, this green group, you can see they are mutant both at 1520 and 1534. Okay, the green group means there are 2 genotypes which are mutant, heterozygous mutant at 1520 and homozygous at 1534 mutant, because the phenylalanine has changed to cytosine, and the other group is mutant at is homozygous mutant at 1520 and heterozygous mutant at 1534, and this red group means this is a wild type genotype, which is homozygous and not mutant at 1520 and also at 1534, right? Oh, are there any chances of metabolic resistance? Let's see what we did. We did synergistic assays. We did 4 approaches. We exposed the insects to PBO only. We impregnated peppers with PBO. We exposed it to PBO and for 1 hour, then we let them to rest for 24 hours. In the next approach, we exposed the mosquitoes to PBO for 1 hour. Then we exposed them to the insecticides for 1 hour, and then we allowed them to rest for 24 hours. In the third approach, we exposed the mosquitoes only to insecticides, which in our case was permethrin, either here or here, okay? And then we allowed them to rest for 24 hours, and we checked the mortality after 24 hours. The results showed as the PBO did not cause any mortality, right? Permethrin caused mortality around 20%. And when we exposed the mosquitoes first to PBO and then to permethrin, the mortality was increased to 100% in Aedes aegypti and almost 50% to Aedes albopictus. Of course, no mortality was caused in the control. In the control, we just impregnated the peppers with solvent just to find out that our assays are fine. Okay, there are chances of metabolic resistance. So let's see the expression of the enzymes which are related to metabolic resistance. So for this, what we did, we isolated the RNA from the mosquitoes, we synthesized the cDNA, then we performed the real-time PCR, and with the help of delta C method, we found out how much is and are these genes expressed. Okay, so the results showed us that the CYP9J28 gene is highly expressed in Aedes aegypti. Okay, in Aedes aegypti, just for your information. And according to the literature, this gene, when it is overexpressed, is only sufficient for the permethrin resistance. I'm going to mention the sensory appendage protein 2 gene, which is highly expressed in pyrethroid-resistant Anopheles. We did this study for the first time in Aedes aegypti, and surprisingly, we found out that it is underexpressed. Okay. Conclusions: Tamifos and pyriproxyfen are still effective, both against Aegypti and albopictus. They are resistant to pyrethroids. Malathion can be used as an adulticide. We could not find any mutations except 1534 and 1520, like 410, 989, 1016. None of those mutations were found, but 1534 and 1520 were found in Aedes aegypti, but they were also absent in Aedes albopictus. In Aedes aegypti, the CYP9J28 was highly expressed, almost 12 times, more than 12 times in Aedes aegypti, suggesting that the metabolic resistance is involved. Here's a summary of what we have done so far. The publication is under process, and the take-home message from here is that the populations are highly resistant to pyrethroids, and they have selected multiple mechanisms, right? This is the first study of its type in Pakistani Aedes aegypti populations, and we are happy. This is my colleagues from Brazil and from Pakistan. And as Dr. Soumya mentioned earlier, that there is a beautiful system of vector surveillance in Lahore. And I'm thankful to all of my Brazilian colleagues. Like, we do all of our things in the lab under the same roof, right from the impregnation of the papers to the HRM analysis and sequencing whatsoever. Some of these guys are directly related to my project, like Louise, I'm going to mention, Barbara here, and Monique, who is recently in France. And my supervisor is hiding behind the cupboard, and the chief of our boss. Everybody was so supportive during my PhD. I'm thankful to everyone. Thank you so much. Thank you, International Society of Neglected Tropical Diseases, to give me the opportunity to talk to you people here. I'm leaving my email here so you guys can talk to me whatever you want. And yeah, any questions, I'm open for it. Thank you, Kamran, if you can listen to me.
Thank you very much. Shukriya both Rafi Saab. That was a fantastic presentation. And in the true spirit of collaboration, Pakistan and Brazil are miles away from each other, but you managed to put such an amazing paper. What really struck us is the robust molecular biology at the heart of it. As you said, it's kind of the first kind of paper of its type from that area. Sorry, excuse me, I just swallowed something. And that's very interesting because only a few weeks ago, we were talking to someone at SciDevNet about giving a voice to the global south, giving scientists a voice in the global south, and this type of thing. So I think that this type of collaborative work in the cutting edge sciences is really very vital to the next wave of innovation that's much, much needed. And I think some of the findings you've got there are going to spark a lot of debate and a lot of further collaboration, and I hope that does involve the private sector as well, which I know a lot of people are actually attending from the private sector. They might be being a bit shy, but we know you're there, and if you're listening, you guys need to get in touch. So thank you very, very much. A round of applause for that, and I'm going to move to the next speaker because of the timing. So thank you, Rafi. I know, and we'll reassemble for the Q&A if we have enough time at the end.
Sure.
So thank you very much. I'd like to welcome to the floor now Dr. John Christian Hustedt from the Malaria Consortium and the London School of Hygiene and Tropical Medicine. The use of pyriproxyfen in control of Aedes mosquitoes. We heard the use of that in the study from Rafi himself as well, and now John's going to talk a bit more about that. So I'd like to welcome John to the floor. John, we can't see you just yet. I think your camera's frozen. I think John's connecting from Cambodia, if I'm correct. I know we've mentioned the Malaria Consortium. I think John's connected. There's John. So I think live from Cambodia. Hello there.
Hi. Yes. Thanks. Sorry, I was having
Brilliant.
some internet troubles. Yeah.
We can see you clearly now. We can hear you clearly. You look great. So
Great.
I'm gonna, under a mosquito net as well.
Yes.
I can see. Yeah, fantastic. Yeah. I'm going to leave you to it. A round of applause, digital round of applause from all the attendees for you. And I'm going to back out now. Over to you, John. Thanks a lot.
Okay. Thanks. Let me just try to share this screen. Let me know if you see it. Right. So yeah, thanks so much for being willing to invite us to share some of the work we've been doing here in Cambodia. So I know that the title was more about our recent paper on pyriproxyfen, but given the short notice, I thought we'd share this, and I added some in there. So it's basically 3 parts of our project that we're going to share today. The first is the trial that we did here with pyriproxyfen, a bit also about our paper, our review paper on pyriproxyfen, and then at the end, a bit on our insecticide resistance study that we did, but it was mainly focused on Tamifas, so I didn't put too much because I thought folks here were interested in pyriproxyfen, but happy to talk with anyone about that. I won't talk too much because I figure probably most of you know about dengue, but just to kind of contextualize things that we have almost half the world at risk, and we have quite 400 million infections every year. And in Cambodia here, majority of the cases are in Kampong Cham province, not majority, but 13% were in Kampong Cham, which is one of the highest rates of any province. We don't have any vaccine or therapeutical treatment yet, so basically we're relying on prevention and vector control. So here's some examples of the different vector control tools that we tested here in Cambodia through the years. You can see that people have tried quite a lot of things, including Tamifas, guppy fish, treated covers, BTI, thermal fogging, and pyriproxyfen. So we did this literature review and went through to try to see which things might be most useful for the ministry here. And we decided to go ahead with using guppy fish and pyriproxyfen in our study, and just trying to understand what places should be targeted. So we looked at, we did these container surveys previously, and found that about 90% of the pupal biomass was in large drums or concrete water jars and these concrete tanks. So if we can get to those, we get a huge percentage of the pupal biomass. And then the remaining, which for all of those you can use guppy fish. And then for the smaller containers where the guppy fish don't live as well, we wanted to come up with something to use in those containers. So we did both. We used our pyriproxyfen in those containers, and we also tried to do some environmental management. Here you can see just another example of that. You can see here these are many containers that we have in Cambodia. You have the large water containers on the top left, and the medium water containers, water jars on the top right, and then lots of kind of miscellaneous containers that we had to figure out how to deal with. We had some study here in published in 2013 showing quite good results using guppy fish based out of a colony that was started from local fish here. So we used that colony that was started by the ministry from local fish here. We had some good results from pyriproxyfen previously, but the product that they had used in the previous study was no longer available. So we used this new product, which is called Similarv 2MR. You can see a picture of it here. And basically, the idea of pyriproxyfen, which probably many of you know, is that you can, if you treat the eggs, you can, it will stop the, will regulate their development and stops them from becoming adults. So we also know that communication is really important, and so we had some guidelines from WHO and UNICEF that came out previously that we tried to model our behavior change interventions and our environmental management strategies around. Here, I'll share just a bit about our literature review we did, looking at the use of pyriproxyfen. So we had this systematic review, and we tried to look through all of the papers we could find, and we ended up including 91 in our study. Out of these 91 studies that we looked at, so it's about 80% were related to one of these four main topics. First being the efficacy of PPF granules, second of auto-dissemination or horizontal transfer, the third the use of pyriproxyfen and ULV spraying or thermal fogging, and fumigants, and then the last being the resistance. So we found that overall PPF can effectively control the emergence of adult Aedes mosquitoes across this wide variety of environments, including granules, ULV sprays, thermal fogging, and fumigants. And utilizing a product with a favorable safety profile is especially important, especially in places like Cambodia or low-income countries where we may not always be able to follow or may not always follow the recommendations exactly. So PPF in granule form results in near 100% inhibition of emergence for 90 days, which has been shown in quite a lot of studies over time. And integrating this pyriproxyfen with other means of mosquito controls can help increase the efficacy and reduce the risk of resistance development. The efficacy of pyriproxyfen may also be altered for populations of mosquitoes known to be resistant, especially for Temmophos. One of the papers in literature were looking at its use with Temmophos-resistant mosquitoes. In areas where the main sources of larval biomass are identifiable and accessible, such as these rural areas where we were in, with these large water storage jars, then controlled release granules or these matrices could be quite effective. But there are other areas where cryptic breeding sites where it's not so easy to reach. So for that, auto-dissemination seems to work quite well. At least for the moment, but there still needs to be some more work done on that to really get a better understanding of the effectiveness of different traps and different formulations. However, even if we did recommend that, there's still no WHO pre-qualified devices that could be purchased for control programs. So that's something that needs to be worked on in the future. And future studies can look at defining the optimal design of these devices. And in areas where Aedes breeding is located in just these large outdoor areas where there's no key containers, and it's not, and you may not want to look for cryptic breeding sites because it's so vast, then maybe the use of ULV or thermal fogging and fumigants might be more useful in those settings. And many studies found that while mosquito populations were still susceptible to pyriproxyfen, the lethal concentrations increased among Temmophos-resistant mosquitoes compared to those resistant strains. And this is even true in areas where pyriproxyfen wasn't really used before. So that suggests probably cross-tolerance of these mosquito larvae to insect growth regulators came up through the extensive use of Temmophos. And so we think that because of that, probably areas where you've already seen increased resistance to pyriproxyfen, they should consider combining the insecticides or changing them over time. Regular entomological surveillance can also help you to understand the susceptibility status and help you have some ideas on how to model your policy. So yeah, that's a bit about our review. I can talk more about in questions if people have more questions, or happy to talk offline with people about that paper. Share with you just some methods of our trial here. We had a cluster randomized design with 3 arms, one with guppies, pyriproxyfen, and combi, and then the other with just guppies and combi, and then the third as a control. And what we thought would happen is that the one that we have, where we had all 3 interventions, would be better, and that the combi activities would help improve communities' knowledge and attitudes and behaviors around that, and that they would we were hoping that those products would be acceptable among the target villages. So we tried to select sites that were far enough away from other places. So we know that in this region, Aedes mosquitoes have a flight range of about 200 meters. So we tried to keep sites which had at least 200 meters buffer between them. And we there and to the cluster. They picked up the guppy replaced for the similar to the mark they needed. The outcome measures were looking very looking at the adult female, resting adult females, Aedes aegypti in the house, and we have secondary outcome measures, other entomological measures, house index, container index, phenotyping, and such. And we did 4 surveys, or every 4 months. We did adult mosquito collection, larvae pupa collection, container surveys, and a PCI. We also did CAP surveys, acceptability surveys, inhibition assays, and coverage monitoring. So here's some of our results. You can see we've recently put up on the preprint server. I can send you a link to if you want to see. We have the some box plots showing that here are the results from the Aedes females, Aedes females by house. You can see that there was significant difference between the guppies, arms with guppies and control arms, especially in the light rain period. So this is the same graph plot for showing Bruto index, same type of results. This is another graph showing pupae per person, also same type of results. And these are our graph, here, our figure here showing coverage, which was quite interesting. Here you see that when you have just one intervention, it's quite easy to keep high coverage of that. So we always had around 80% coverage of guppies in one arm, but in the arm that had the combined interventions, when we try to emphasize the use of one intervention, the other one, the coverage of the other would go down. And in other reviews, we've seen this has been consistent with other vector control tools as well. So this is kind of important to think about if we're wondering what kind of policy we want to make. Then we have this last figure here, which is just showing that people knew about dengue, they knew about Aedes mosquitoes, where they were, they believed they could prevent dengue, but they didn't know, they didn't have much knowledge on using larvivorous fish. And key messages that came out of this trial was that dengue control in Cambodia is still difficult and relies mainly on vector control. And we see the effectiveness of both guppy fish, pyriproxyfen and combi was quite well, but there are still places where the intervention, the areas where the intervention was deployed had significantly less pupae and larvae and adults. And the guppy fish were widely accepted and used by people in the intervention villages. We know that community engagement is really important and led to people wanting to use them and having high coverage. And when we went back to look at coverage a year later, still many of the villages and some of the control villages were using them. So for pyriproxyfen, part of the problem is that that product is not for sale here in country, so people haven't been able to get replacements, and we still don't know if they would purchase them or at what cost, or if the government's going to purchase them, if people would continue using them or not. Some of the limitations of our study here is lack of epidemiological endpoints. We had hoped to do that, but we weren't able to. We need a longer-term evaluation, see over 5 years or so, see if people are still using those products or not. We do have a few of the pyriproxyfen left for those intervention villages to keep using to see if they'll use them. We didn't target public spaces like monasteries or parks or things like that. So that could be important in future studies. And we also need to understand how to control that biomass outside those big containers, maybe auto dissemination or other ideas. And then here's just a quick bit about our paper. If you want to look it up, you can see the link here. This was looking at the resistance of Aedes aegypti from around the country. We collected from 4 different places across the country and looked at resistance to deltamethrin, permethrin, and temophos, and we found that there was quite a lot of resistance in temophos, well, in all three. If you have more questions about that, or you want to read our paper, or happy to talk about that. In future projects, we are looking with WHO TDR money. We have a follow-on project here, which is nearly complete, and results should be coming soon, which looked at the use in schools of guppy fish, homemade mosquito traps, and working together with the Ministry of Education on a curriculum for students to teach them and hopefully they go home and teach their parents. There's another trial called My School, which we're working on in Myanmar, which is using guppy fish and also domestic larvivorous fish, and doing some studies there to see which types of fish there might be most useful. Looking at homemade mosquito traps as well, and working with Ministry of Education and Ministry of Health on behavior change and environmental management. And the My School trial has quite a large sample size with epidemiological endpoints, which should be nice. I'd just like to acknowledge our different partners here. We have our CNM, which is the government partner, the Ministry of Health, WHO, US NAMRU Group, London School, Sumitomo, and our funders, GIZ and UKAID. And yeah, so that's it for me.
Thank you very, very much, John, for that. We loved your guppy fish biocontrol. In fact, we interviewed you guys for an article that we published about 1.5 years ago as part of the ISNTD press corps when the first set of results came out. And then Prudence Hamad came in person to discuss the combi framework that was being used at one of our meetings. Actually, the video of that's up in our on the archive on the YouTube channel that we've got, which is open to everybody. So it'd be very interesting to kind of expand on that in the Q&A with you a bit. And I really would like to learn more personally about the school project in dengue. We've ourselves set up, I'm sure we learned from you, an outreach program for schools for dengue outreach in southern Tanzania. It went live last weekend, and we had an ISNTD Connect session with the on-the-ground people that are running it there. Maximilian Godwin is one of the people who are running it for us there in Tanzania. So I'd love to learn more about that. So perhaps we can exchange some ideas on that. So thank you very, very much for that. We really appreciate that. You've done it undercover in terms of the mosquito net, which I can only take my hat off. And I can take my hat off now because I've had my hair cut. So you should have seen it yesterday, but that was fantastic. Absolutely love that. A lot of different things going on in there. So I'm going to say thank you very much, digital clap, and then move on to the next speaker. Thanks a lot for that, John. And I'm sure a lot of questions, there are a lot of questions coming through from the audience as well. That kind of takes us to the next speaker, old friend of the International Society for NTDs. We've had the pleasure of having Dr. Beth Poulton come to our SciComm. We run a science communication festival every year at the Wellcome Trust. This year's was the 5th annual one. Beth came to the first one 5 years ago. We did it at the Barbican, that particular one. Ever since then, we've been at the Wellcome Trust after that. And that's looking at, we give awards for apps, print, comics, films, television programs. You should see all of that on our YouTube channel. And please do subscribe to that. It's free. There's 8 years of video archives, presentations, interviews from our film team, and our we have 5 meetings a year, live physical events. You can see all of that on the YouTube, but you have to subscribe to it. So I'd suggest that's a great resource for you. There's some absolutely amazing stuff on there. And that kind of brings me to Beth. The automated phenotyping of mosquito larvae enables high throughput screening for novel larvicides and smartphone-based detection of larval insecticide resistance. Something that John kind of said in one of his slides, there was a line about keeping entomological surveillance regular is needed to monitor resistance. And I'm just wondering what a tech solution could perhaps bring to that. And so I think that's a nice segue into getting Beth on, if that's possible. And a round of applause for Beth. Yay, digital round of applause. And I'm going to back off and let you do your thing. So see you in a second. Thank you.
There we go. Thank you very much for having me, Kamran. I'm very grateful to be here. What I want to talk to you about today is some research that we've been doing over the last couple of years with LSTM, UCL, and the London Centre for Nanotechnology around developing a high throughput assay, which allows us to rapidly assess new larvicides, detect resistance, and we've also now got a smartphone version of the system, which has a lot of potential. So last week we released a preprint on bioRxiv of this work, if you would like to go and look at it a bit more in detail. So the information's there if you'd like to see it. We've spent all morning talking about viruses like dengue and other mosquitoes like Anopheles, so I'll not dwell on this. I'm sure you all know that mosquitoes are bad. I'm sure you're familiar with the life cycle, but just in case you're not, adults lay eggs into the water, which hatch into the aquatic larval form, emerge into pupae, and eventually emerge as an adult mosquito. So for decades, adult mosquitoes have been controlled very effectively, particularly for malaria, using long-lasting bed nets and indoor residual spraying using pyrethroid insecticides. However, problems have arisen as not all mosquitoes bite indoors, particularly the Aedes mosquitoes, which spread viruses like dengue and Zika. And there's now widespread resistance to the pyrethroid insecticides that are typically used on in these methods. One method which we can use to combat this is by controlling the larval stage, which is widespread for Aedes control and typically involves either removing or covering the water sources or treating the source with insecticides or with biological agents such as BTI. As we're using when you're using a compound, there's always the potential of resistance developing. So it's important that we can monitor and keep track of how susceptible our population is to the insecticides we're using. The WHO will have a standard assay for doing this, which involves putting 25 third instar larvae into a plastic pot around 250 ml. With 200 ml of water plus varying concentrations of your insecticide. And then after 24 hours, checking to see how many of the larvae are still alive. This is a quite reliable assay. It gives nice graphs where you can quite easily often see that there's a difference in susceptibility between two strains, which is very useful. However, as it's third instar larvae that you need to use, the rearing requirements are quite laborious. You need to rear lots of larvae to do this. It's time consuming to actually set up the physical assay. As a result, it tends to be low throughput. Also, the bench space required tends to be a limiting factor to how large assays can be set up. And finally, there's an investigator bias, as although there's quite strict rules around what counts as a dead larvae, there is a bit of personal opinion in whether a larvae is alive or dead. In light of this, the team at UCL, partnered with LSTM, have developed a high throughput assay, which reduces the rearing time, is far more high throughput as it uses 96 well plates. It reduces the bias of data collection, as the collection is done by a camera and then a computer algorithm. It's much faster and far less physical space is required to conduct this assay. This assay involves using the Invertebrate Automated Phenotyping Platform, or INVAP for short, which was originally developed by the Satel lab at UCL to analyse nematode motility, but we have now modified it to use with first instar mosquito larvae. So 3 to 10 first instar larvae are placed into each well of the 96 well plate. Prior to adding insecticide. Insecticide then added, incubated for the time period desired in the experiment. We've typically been using either 4 or 24 hours, but any time period in between can be used, as it only takes a matter of minutes to collect the data using the camera. And short movies at each time point are taken. The basic concept of this assay centres around the idea that mosquitoes that are moving are still alive and haven't succumbed to the insecticide. So as Kamran was mentioning, a bit of simplicity in these kind of things is a really big benefit. So in terms of the analysis, this is completed in Python. So movies are imported into the script. Each image is divided into a 12 by 8 grid, as 96 well plates always come in 12 by 8. So each well has its own little segment, which is analysed on its own. For each well, the variance between each of the images in the movies is calculated using because there's a change in the colour which can be detected, and if it's above a certain threshold, it's counted. So we're able to see how many of the pixels change in each well across the movie, which gives us the movement index of how much the larvae are moving in that well. And then we normalise that movement index. So that comes back to the movies that you take prior to adding insecticide. So this is important because the larvae are biological creatures, they're not identical, they're not clones. So it's important that we account for the number of the differences between larvae, so that we aren't just detecting a natural difference in the amount that larvae moves, and it's not actually a difference as a result of the insecticide concentration. So INVAC is a very versatile system. It can be used for lots of different things. One of the things which is described in the preprint I mentioned before is screening large compound libraries for lethal activity. So we've screened the Medicines for Malaria pathogen box, which contains 400 drug-like molecules. And we screened all of these at a set concentration and identified 5 compounds which showed a level of insecticidal effect. We then did concentration curves for each of these compounds and identified the compound in the bottom right-hand corner of the graph there, which turned out to be tolfenpyrad, which is an established pyrazole insecticide. We also identified clemastine, which is the drug on the bottom left, which is an antihistaminogenic drug, which also showed a paralysing effect. This is just the first step in using this to screen for new compounds, and it has a lot of potential to be a very high throughput assay to potentially identify mosquito lethal compounds. Another thing this system could be very useful for is for resistance profiling. So by using a concentration range and then comparing to a susceptible comparator strain, we can calculate the level of resistance present in the population. So to do this, we generate a generalized linear model of the movement index against concentration for each insecticide strain and each insecticide grouping that we are looking to analyse, which gives us nice sigmoid curves as shown here. We then calculate the inhibitory concentration required to reduce the movement index to 0.5, which we can then use to calculate a resistance ratio. So it gives us an idea of how much more insecticide is required to reduce the motility by 50% in the resistant strain compared to the susceptible strain. And what everyone's probably quite excited about now, the smartphone assay. So the group at the London Centre for Nanotechnology has been crucial in developing this, and it involves the same algorithm as is used in the INVAC system, but it's completed on a camera phone. So we still at the moment have been using 96 well plates, and have then, rather than doing a concentration curve, at the moment we've been using a single concentration and filming continuously for around 200 seconds. In that time, what we're able to see is a difference in the length of time that it takes for the motility to reduce for susceptible versus resistant strains. So as this is a smartphone assay, it has potential to be used in the field. It could potentially permit geolocated data collection on resistance, and we've shown it so far to be successful in detecting resistance in both Aedes aegypti and Anopheles gambiae to deltamethrin. So it has quite a lot of potential and has potential to be very simple to use as an app on the phone without requiring coding ability to actually use it. So there's lots of potential in the systems that we've developed, and hopefully they can be further developed, and with the increase in the quality of camera technology and the decrease in the price in order to buy good quality cameras, it probably could be quite a cheap assay to actually have in the future. Just before I finish, I'd like to thank my supervisors, Gareth at LSTM and David Patel at UCL, plus all of the other people that have helped on this, particularly Steve Buckingham and Freddie, who helped develop the system, and my colleagues at LSTM, Amelie and Fraser, and the team at London Centre for Nanotechnology, Dr. Miller and Professor McHenry, and my funders, the MRC. Thank you.
Okay, sorry for that. I just had a bit of a delay getting onto the mute thing. Beth, thank you very, very much for that. What's there not to like about INVAP? You know, appropriate technology at its best. I mean, I think you're going to get a lot of inroads in terms of people looking to come to you for that. The London Centre of Nanotechnology, I mean, for me, that embodies the actual collaborative ethos. I remember when it first opened just next to UCL. I remember that when it happened. I remember the people involved in it as well. Fantastic multi-sectorial hub of creativity, actually. If anybody's in London, you should go and check that place out. It's quite intense, actually. Little door. I know it's a fantastic place, just around the corner from the London School of Hygiene and Tropical Medicine as well. So a fantastic show of collaboration. And thank you very much for that, Beth. Fantastic to see all of that. What I wanted to say was that now I think we've got I'm just trying to get the timings up, sorry. Yep, this is due to end at 16:05, so we've actually got time for a bit of questions, a few questions from the audience. I'd like to, if that's okay with the panelists, just reassemble for a second. And I'm going to, we talk about collaboration on this particular chat. We've got Jackson from Nairobi, Elton from Albania, Franklin Assisi from Cameroon. We've got Ahmed Awais again from Ghana, Marie from Paris, Shahid from Pakistan. We have Kenya, Emily from Kenya, Faith Folahan from Nigeria, Stephen Bramia in Canada. We've got a lot of people coming through from Brazil. You've got a big fan base there, Rafi. So Jose Roberto, mashallah. We've got Dr. Assistant Professor Mohammed Tariq from the Entomology Arid Agricultural University of Rawalpindi in Pakistan. Some great, country spreads there. And some of the questions that have come through, Faith, well, there's a statement of Faith is saying that research, great job, Rafi, research of this kind is lacking here in Nigeria. Your work is an eye-opener. There's a question here, I'm coming to that in a second, and that's from Stephen Bramia. I'm just trying to get it. Second. There's one from Ademir saying, Rafi is a brilliant student, a very good friend. Thanks for choosing these four. I'm just trying to find the question on this scroll. Just bear with me 1 second. Yes. So this is interesting. Jose Roberto has mentioned that it'd be very interesting to assess the role of the microbiota in all of this, in the sense that he's saying that microbiota plays a very important role in some of the physiological processes, but there's not been much of a focus on that. That's very interesting because Dr. Alvaro Acosta Serrano, London School of Hygiene and Tropical Medicine, we had an ISNTD Connect with him a few weeks ago about leishmaniasis, and in that was a lot about the microbiota of the gut, the fly's gut, and how that can influence issues, metabolic issues. And your work was on these metabolic kind of enzymes, and perhaps that's something. Is that an angle to pursue? Do you think?
Is that for me, right?
That's for you, Rafi. Yeah.
Okay. So yeah, my lab is particularly working on the the things that I have told. I have used all of the resources of my lab to tackle insecticide resistance in Pakistani populations, but we have a lab right close to us, and people are working there with the microbiota. And Jose knew some of them, like there is the group of Rafael, his colleague, Dr. Mariana. She is doing some kind of work with the microbiota, and I'm not sure if she is putting any kind of light on if she is like correlating a microbiota with insecticide resistance. But I believe there is something going on about microbiota and insecticide resistance. So it is an interesting topic. We can do it, like, for example, it's not really complicated. We can compare the microbiota of the susceptible population, and we can then find out the microbiota in resistant. So we can find out what is present, what is the microbiota of the susceptible population, and what is the microbiota of the resistant populations. So I believe there are people working on that, but our lab is strictly, to this area, because we are working with this impregnation papers. My professor can comment better. We have a collaboration with World Health Organization, because this impregnation of papers is a big issue. People import them from Malaysia, and arriving from there, and all those complexities, logistic problems. So we are trying to recognize our lab as one of the centers of paper impregnation in South America, so we can do that. And the molecular and the biochemical aspects of insecticide resistance, I think our lab is, because I am from this lab, so I think it's the best lab. And you saw my study. We have used all of the resources that we have in our lab to tackle the insecticide resistance in Pakistani populations. I'm thankful to my professor who let me utilize all of the resources that we have in our lab to do my studies. So yeah, this is interesting, and I think there is something going on about microbiota and insecticide resistance.
That's fantastic. So there's room for thought there a bit. I mean, and we've got a question here that's come through from Stephen Bramia for John. Firstly, he's saying, Stephen Bramia from Canada is saying kudos for the guppy work, and also he's asking, are they inert to PPF?
Yeah, so he's talking about the guppy fish, right? Can they live in the same? So yeah, so we also had some of the containers. We put the guppy fish in the containers with the pyriproxyfen, and they seem to survive fine. So that's more than 2 years now. We put the recommended dose in the large water containers at the health center, and and the guppies surviving several generations now.
Okay, hopefully that answers your question. Someone I think has come back with that. Okay, that's fine. Derek is asking. Derek Charlwood is asking, is there a video of the use of the phone available, Beth? Does the kind of 96 well plate affect the results? So the high throughput.
I should be able to get a video of the smartphone. I've got a video from the INVAT system, but not personally the smartphone, but I'm sure I can get that if someone wants to look at it. In terms of the type of 96 well plate, it shouldn't have a massive impact, but one thing that you have to be wary of is if it's not completely straight edges, the camera looks down on the 96 well plate. So if it's got a bevel on the bottom, the larvae can end up out of sight of the camera. So it's better to have completely flat bottoms plates to prevent the difference in terms of the way the camera sees the plate. So the larvae can't hide around the sides.
And in terms of scaling, thank you for that answer. In terms of, I hope that answered the question there, Derek, for you. In terms of scaling out that approach, the INVAT, different countries, is there a plan for that? What obviously there is, but what are the challenges around that? Are there kind of hurdles around that? Because it seems a very appropriate technology that would address issues such as the one that John raised on one of his slides in terms of keeping entomological surveillance regular, in terms of using that to then monitor resistance and regularity and consistency. I think you've answered those two questions with your approach with INVAT. What are the hurdles to scaling that out?
Yeah, at the moment, the system on the phone isn't quite an app. It still requires quite a bit of input from the researchers. So there's work going on at the moment to develop that so that it is a complete app that can be used by someone without a lot of statistics knowledge. But it is being worked on behind the scenes. This is just the first paper to come out. We have quite a lot of work coming out in the next year or so of developments and different uses of the system.
Yeah, and presumably part of that development curve, you'd be pooling that data so that eventually the policy, the famous evidence-based policy heads can actually dip into this kind of that stream of data. Is that you'd be looking to clouding it, pooling it?
Yeah, so I have some work that I'm working on at the moment that I hope to get published before the end of the year, looking at genetically modified strains overexpressed in metabolic enzymes, in the hope that I've screened against lots of different compounds, in the hope that it can then inform policy to suggest if you have a particular metabolic enzyme overexpressed, you can't use these insecticides, but other classes may be an option.
So I'm sat here thinking, why don't you three do something together? Because I think you'd probably benefit from that, John, in terms of your, in terms of this regular monitoring line that you use on one of your slides, definitely. Rafi, you're involved with this metabolic enzyme overexpression, or at least the differential expression of it, and mapping it to resistance. And then you've just heard that from Beth. I hope something good comes out of it. I hope something happens. That's what this meetings, and that's what our ethos has always been. Do you think there's any mileage there, or are we barking up the wrong tree?
Potentially, I'm sure we could have a chat and see whether there's any collaboration possible there.
Yeah, you never know, people. Any thoughts? Any thoughts on that, guys?
Kamran, I have been to this first Congress of the World Insecticide Resistance Network, which took place here in Rio de Janeiro, and it was amazing. You could see how people can work together. People are working in Japan, people are working in Australia, people are working in China, India, Brazil, Africa, US, Canada, and how when they work together, it has an impact,
Absolutely.
So the problem, I don't know what happened to World Insecticide Resistance Network. They had another meeting in 2018 in Singapore, and after that we could not see any other activity about World Insecticide Resistance. But as you mentioned, if we work together, there is clearly an impact, and all these activities they must be streamlined so that the people can get, we could show the people that this is the fruit of our work that we are doing together. As you mentioned, there is a link between all these things, but the link is not connected.
Yeah, I agree. I totally agree with that. John, what do you think that connection is? Is it funding? I mean, is it what is it? You're the Malaria Consortium. We know, Charles Nelson. I've met Charles. You're in person in your offices here in London a while ago, and the Malaria Consortium were actually on our dengue advisory group. Michael Haydock, who's just left, he was the latest person from the Malaria Consortium, and now we're looking to get another person from the Malaria Consortium on the dengue advisory group. I think it was Sophie who was there before, who'd already left. In terms of this conversation, what do you think that hurdle is?
Yeah, I mean, from my perspective here, I think, or in any of these kind of resource limited settings, I think any kind of advantages you can have, like what the tools that Beth is putting together, I think would be really useful. Definitely here, it's not only money, it's also manpower. So there's only so many people that have the expertise to be able to go around and do these studies, and there's only so many facilities available that we could do them at. So the kind of the quicker we can do them, the cheaper we can do them, and more at scale. If the things that Beth puts together on her team, then definitely it would give us a lot more data and a lot more opportunity to try to influence policy in the right direction.
Yeah, I mean, it's very interesting that you say that because this morning when I opened the first session, I mentioned the 3 paradigm shifts of the WHO's NTD roadmap to 2030. One of the major seismic shifts is they're trying to foster this movement from the program perspective. From a international donor-led external stakeholder model to a far better sovereign finance, country-led ownership model. So there's a lot of and you mentioned getting the finances and getting these things scaled out to different countries. There's a rather gap trying to inform governments and the policy people that have got the actual money in the endemic regions, in the countries themselves, that this is beneficial to your actual GDP. So I'm trying to think, is it worth perhaps one of the missing links in the stakeholder mix are the health economics outcomes people build an economic case. In dengue, we had it with people like Professor Don Shepherd, who was putting out some amazing work in terms of what happens, Bruno Guy, loads of people looking at the economic case for if you have this intervention, how it can benefit that country, and that ends up triggering funds being released by that country and pulling together the science-based different communities of scientists. So is that something to think about, perhaps? The socioeconomic case for what you're working in one silo, but the actual impact of it perhaps should be measured in a different way if you're looking at a larger, wider collaboration. Yeah,
definitely. I think the more I think that the more data and the more information that policymakers can have to make the decisions, I think is good, and also for donors, because it's not just governments, but oftentimes governments will or donors will want to purchase something, and then the countries will have to go along with what the donor wants in some cases. And I think that if the donor, sorry, my light just turned off. If the donors they want to make a recommendation, it's important for them to have the right information.
Yeah.
And then they make proper recommendations for this for the policymakers as well.
Fantastic. I'd just like to say thank you very, very much. We've got a minute or so left. Does anybody else want to add to that? The last bit, Rafi, you look like you're about to say something.
Yeah, there is a question about the use of insecticides by the common people, and there is a very interesting study about the Sao Paulo state in Brazil that the government has stopped using deltamethrin, but the insecticide resistance is still increasing, So there was a question: if the government campaigns are not using deltamethrin, why is the insecticide resistance increasing? And when they looked into the problem, they found out that the people are still using insecticide resistance as household, sprays or repellents or whatsoever. Not repellents, but household insecticide sprays. So this is a big issue, and we need to educate people about that. Like in the previous lecture, they talked about 10 minutes against Aedes in Malaysia or Indonesia or somewhere. So the same thing is taking place here in Brazil, and the name of that is 10 minutes against Aedes, where we go to the schools or public events and we educate people how to eradicate the breeding sites of the larvae of Aedes and all mosquitoes. And we educate students and kids and people. So we are trying to bring those stakeholders together, like the common people and the government, and we want to take our lab research into the field and then tell people what's going on.
That's very interesting, because there's Faith has come through on the chats there. There's quite a few questions here, and she's asked a general question, firstly from a personal, echoing what you're saying, that she's observed the issue of insecticide resistance is known commonly to scientists, but not to the majority of the population. So there's this kind of gap between the science base, who are very aware of it in the metabolic, up to that kind of level, as you've just explained, the nitty-gritty of it. However, the the population general are completely unaware of it. And she's saying there's no adequate publicity or education to the general population about this problem, and lots of them still use these insecticides. She's speaking from Nigeria, so the example she's giving as Nigeria, she's saying, my questions are, who should take responsibility of stopping the use? That was actually, I was going to ask that. Is it private sector? Is it public? Who is the manufacturer? Who? And secondly, what is the effect of consistent use of insecticides that are ineffective on further mutation of vectors? So whose responsibility is it to educate the public, bridge that gap? Secondly, are there any effects on long-term use in terms of selective mutation?
I think in terms of responsibility, there's kind of a level of responsibility on everyone, in that scientists have a responsibility to learn about it, actually have evidence for why resistance is developing and what we can do to stop it. Because it's very hard to go to governments and say, you need to stop using that one without evidence to actually back that up. And then governments have, and scientists together have a responsibility to then actually educate the public and explain to people and really do a good job of presenting the evidence to kind of non-scientific people. Yeah. Because people don't willfully ignore things. There's reasons behind it. So if explaining the evidence doesn't do the job, we then need to actually look at what are the reasons behind people choosing to still use deltamethrin.
Yeah.
Even though they've been asked not to, there may be a lot more to it than just people ignoring like what they've been asked to do. In terms of the effect of using them consistently, it can cause resistance to become more strong, particularly with metabolic enzymes where you get cross-class resistance.
Exactly.
There's the potential that a low level of resistance won't have an impact on other classes. A low level of overexpression, won't have an impact on other classes to the same extent as a much higher level of overexpression will.
That's a fascinating area to open up. In terms of probably another session is needed to go there in terms of that, because there's a lot of that, and that's a fantastic question by Faith from Faith Folahana from Nigeria. John, I don't know if your screen's frozen or you're looking like Jim Morrison on the cover of LA Woman, but it's I don't know, it's the way the shadows are playing. We have to I'm getting told that I'm being given the death stare. I will actually turn the computer around and show you what I have. You don't know. We've gone over the time. They're going to kill me. I'm going to have to say thank you very, very much to everybody. Firstly,
thank you.
Yeah, thank you, Beth. Thank you, John. Thank you, Rafi. Delightful conversation. And I think it'd be really good for the audience to continue this. There are chat rooms. There's quite an immersive platform. You can ask for one-on-ones in terms of furthering research collaborations, and that goes out to the audience as well. And this will be live, and this platform is there in perpetuity. You've got it forever. Anybody can drop in and out, not just today, but forever. We'll be posting stuff. You're welcome to post stuff, papers, videos, anything. It's there. So thank you very, very much. And I think we can leave it there. Okay, so round of applause, virtual applause again. Thank you.
Thank you.
Stay safe wherever you are. Stay safe wherever you are.
Thank you. Assalamu alaikum, Kamran.
Walaikum assalam wa rahmatullah.
Thank you.
Rafi, Eid Mubarak to you
Eid Mubarak.
in advance, and enjoy the Qurbani and everything. We're doing it here as well. So
thank you.
You and your family and everybody. John, keep cool, safe in Cambodia under the net. Don't let them bite. Beth, take care. All right?
Thank you.
Thanks a lot. Bye-bye, everybody.